Hey everybody, welcome to Professor Long's Lectures in Anatomy and Physiology. As you guys know, we're in a coronavirus shutdown. I hate saying that every video, but I want to preface this because some of these videos may stay on YouTube for a while. And I'm going to try to redo all my videos when I get the opportunity, but right now we are in a rush for time to get all of our class switched over to online from traditional face-to-face. And I'm doing a bunch of impromptu videos with a minimum of equipment, no editing or anything. So warts and all, this is what you get. I hope to redo these in the future, but right now we're just pressed for time. So this lecture is intended for my 2401 AMP1 class. This is the second lecture in the series on the nervous system. The last time we went over the organization of the nervous system and sympathetic, parasympathetic. I will ask a number of questions on your quizzes and tests about that, about afferent versus efferent, autonomic, sympathetic, parasympathetic, what controls what, interoreceptors, exteroreceptors, all that stuff. Now, the next lecture I'm going to do is going to be talking about neurons and the anatomy and structure of different neurons. I'm not going to go through all of them. I'm going to go through the basic ones that we need to worry about for right now. And then I may get into the action potential. It depends on how long this video runs, how much I'm going to get done. Okay? So, when we study the nervous system, there's two types of cells found in the nervous system, two basic classifications of cells. One classification of cells is called neuroglia, or some people say neuroglia, either way. And sometimes we just say glia. Neuroglia or neuroglia are cells that do not do information processing per se. These are sort of the support system or the support cells for neurons. And we're going to go into detail of the neuroglia later on in this series, but they're one class of cell that provides sort of a physical, structural framework for neurons and do some other things. The other type of cells in the nervous system are called neurons. Neurons, they do the information processing. This is really the functioning part of your nervous system, although glia play a super important role in how neurons function. Our neurons come in different shapes, different sizes, and different morphology. Morpho refers to shape, and so the morphology of neurons can be extremely varied. As a matter of fact, as an undergraduate, I worked in a cell morphology and a neuron morphology lab, and we were studying the shape of certain neurons in the hippocampus of the brain and looking for synaptic connections and doing all sorts of really cool stuff. I don't have time for that now. I'm going to look at neurons. I'm not going to worry about neuroglia right now. Neurons can look different from each other, and they're tremendously different from other cells. Neurons fall under a class of cells called excitable cells, and there's two types of excitable cells that we've talked about, or we're going to talk about: muscle cells and neurons themselves. We talked a little bit about neurons in the last series of lectures working up towards the skeletal muscle and its function, because we had to talk about motor neurons. We didn't talk about how neurons function in great detail, though. So when we look at neurons, most cells are just either round or cuboidal or columnar or whatever. Skeletal muscle cells are long cylinders. Neurons have many, many branches coming off of them. So in typical, we have a number of different shapes of neurons, and I'm going to talk about the shape of each one of these neurons that are important for us. There's many more than this, but these are the ones we'll talk about. Just like any other cell, neurons have a nucleus where all the DNA is stored and codes for all the proteins and blah, blah, blah. Have all the normal organelles that we typically see in most cells. And then some neurons will have all these little branches coming off of them and going in many different directions like this. And when I look at this neuron with all its little branches sticking off, none of the branches are quite defined in the way that I'm going to define the branches on these other neurons. So when we talk about neurons, neurons can have two major branches, or types of branches that are really common. One group is called dendrites, and the other one is called axons. The dendrites, dendro means branches, and usually these guys are considered the dendrites of this neuron. This neuron doesn't have a really long branch where it goes and communicates to another one. So a lot of times these are called anaxonic neurons. An meaning no axon. Okay? I'm not going to talk a lot more about anaxonic neurons. I drew it for you. We talked about it. And you'll see what I mean when I talk about axons when I draw this next one. The three basic types of neurons that we're going to use and talk about it quite a bit is going to be this. Some neurons are called unipolar neurons. And by the way, in that last drawing, the round part where the nucleus is referred to as the soma or cell body of the neuron. So when I look at a unipolar neuron, it's going to have a nucleus. It's going to have, all the typical structures that a cell would have. And then off one side, it's going to have this really long branch. It's really kind of one long continuous axon that'll have some branches at one end and some at the other. Okay, and it turns out that neurons are usually one-way streets for information to travel down. The information that's going to travel down a neuron is called an action potential. It's electrical voltage that runs down here. And neurons are always a one-way street. If these branches would be called the dendrites, and then this is the axon, and at the end of the axon, we're going to have these smaller branches. They are called telodendria. And then at the end, we would have synaptic knobs. Now, we talked a little bit about this when we did the neuromuscular junction. The synaptic knob, also called a synaptic terminal or synaptic button. And so when we look at a neuron, this neuron is called unipolar because off of the soma, this part appears referred to as the soma. Soma means body or cell body. Off of the soma of the neuron or off the neuron cell body, there's only a single branch, a single pole. And it's a very long continuous axon that has some dendrites and some telodendria at either end. The next type of neuron we can look at is going to be called a bipolar neuron. In a bipolar neuron, we have our nucleus, we have our soma. But off of this one, there's going to be two poles or two branches. One long branch like this that can have a bunch of little smaller branches at the end, and another longer branch down here that will eventually branch off into telodendria and synaptic knobs. This would be called the dendrite. This would be called the axon. And this is the soma of the neuron. So in bipolar neurons, we have a single dendrite and a single axon, each coming off of the soma on opposite sides. It's got two poles. It's bipolar. Has nothing to do with bipolar disorder, which does have two extreme poles, either really manic and excited or really sad and depressed. And we're not going to talk about that right now. So now, the last type of neuron I'm going to talk about is called a multipolar neuron. When we look at a multipolar neuron, they have multiple poles or multiple branches. And really in a multipolar neuron, what they have is they have lots of dendrites, many dendrites coming off of their soma, and then a single axon coming off that's going to split into a bunch of telodendria and synaptic knobs. And these dendrites are highly branched, extremely branched structures, and they're absolutely amazing looking when you see pictures of them. I would encourage you to look up a multipolar neuron image or a dendritic cell image on Google or whatever your search engine is, and you'll see these beautiful three-dimensional cells. Anyway, this is called a multipolar neuron because I have many poles, branches coming off. All of these are dendrites. They have a single axon usually, and the soma with the nucleus and all the organelles is in the middle. Okay. So you can see why these are called multipolar neurons. Now, a lot of what we're going to talk about and a lot of the work we've taught that's been done on what we're going to talk about as far as conducting action potentials and things has been done on multipolar neurons, the giant multipolar neuron and the giant squid axon. And all the other neurons function almost identically, but there's some minor variations, but ultimately the theme is the same. So I'm going to worry about multipolar neurons. I'm going to draw the anatomy of a multipolar neuron, and then we're going to talk about how they function. Okay, so I'm going to erase all of these. You do need to know anaxonic neurons don't have any axons. Unipolar neurons have a single long continuous axon with some dendrites on one end and some synaptic knobs on the other. Bipolars have a dendrite and an axon, and then some little branches at one end of the dendrite, some branches at the end, but they are bipolar, two branches off. The soma lies in the middle. And then multipolar neurons have many dendrites coming off the soma and a single axon usually. Now, that's not always 100%. There is some occasions when you look at neurons where they can have a second axon branch off And anytime there's other axons running along, they call that a collateral, which means with the side. Axon collaterals are additional axons that can run next to the axon of another one. But it's not super common, and we're not going to talk about collaterals beyond that. Now, I'm going to erase all this, and I'm going to draw you a multipolar neuron, and we're going to label it. And I know we've done this in lab, but I'm going to do it anyway, so that when I do it in the next video, we're actually going to throw ion channels on here and see how action potentials occur. So now, if I had a multipolar neuron, I'm going to draw a small one here. There's the nucleus and the soma. Here's the axon coming off with all the little branches, multiple telodendria at the end. But then I would have all these massively branching dendrites coming off the soma. Okay, many of them. If I were to try to draw one larger, I mean, this would be the size of a tree that's in your front yard with all its branches. I mean, they're really huge and multi-branch structures. So what I'm going to do is I'm going to trim all these down, and I'm going to draw pretty much a few dendrites, the soma, and all of this, and we're going to talk about the parts of the neuron. Okay, so here's a dendrite that's been cut. Here's another dendrite that's been cut. Bear with my silence here for a second. So this isn't the prettiest neuron, but they're actually not always very pretty like the pictures in the book show. In here would be our nucleus. All of these branches out here would be called the dendrites. Everything, if I could kind of draw an imaginary circle here, or a dotted circle here, and trim all the branches off, this part all the way across here, that the dendrites and the axon are attached to, is called the soma, or the cell body. And if I wanted to say plural, the word for plural for soma is somata. We don't say somas, it's somata. So anyway, so the soma is the neuron cell body. And then attached to the soma, what connects the axon to it is this area that tapers down called the axon hillock. And some people just call it the hillock, but it's the axon hillock. This would be the axon. These little tiny branches at the end, as you should know, are telodendria. Telodendrion would be singular, telodendria would be plural. So it's a telodendrion, and then we have the synaptic knobs, or the synaptic terminal, or synaptic button. I like to call them synaptic knobs because they look like doorknobs to me. And even in French, they're called synaptic bouton. Anyway, la croissant. So this is the basic anatomy. There's a little bit more to it, though. Now, inside the soma, we usually have a lot of the organelles, endoplasmic reticulum, Golgi apparatus, ribosomes, and all these other things. The first guy to be drawing these out was actually a pretty young guy. There's a great book about him. I forget the name of it, but it's based on a gentleman who lived in Spain who was actually a young teenager named Santiago Ramon y Cajal. And you can look him up. His last name is Ramon y Cajal, K-A-J-A-L. Santiago Ramon y Cajal was working in his father's lab, from what I can recall from the books. It's been a long time since I read this stuff. But I believe his father was a physician, and he was working around the doctor's office and taking some tissue from some brains that were in jars and slicing it up and looking at it under a microscope and studying it. And in order to see the neurons, he had to put it in certain stains, and took these thin slices, put it in these stains, a lot of silver stain, and he saw these beautiful shapes of neurons in the tissue under the microscope, and he started drawing them all. His drawings are very famous. So he started naming a lot of these terms and describing the neuron. Still didn't know how they functioned, but at least he knew there were cells there. And one of the things that he realized was that we didn't know this was a nucleus. It was called the karyon. Karyon means kernel or seed, because he saw this little ball that showed up inside there that always stained sort of this dark color. Didn't know what it was. We didn't know it had DNA or what its function was back then. This was in the late 1800s. And so he called it the karyon. And it turns out that there was an area surrounding the nucleus inside of here that stained really, really, really darkly for some reason, and it made a very dark band that showed up around the nucleus. It's just kind of a little cloudy area there. And they called that area the perikaryon. Peri means around, and then karyon means the kernel or nucleus. So the dark staining area in a neuron around the nucleus is called the perikaryon. Now we know that there's all sorts of organelles that are picking up that stain and are concentrated in there. There's even a structure called Nissl bodies, Nissl. You can look up Nissl bodies if you want. I'm not going to be too concerned with them. Now, the reason this becomes important is because how neurons function is neurons function in order to carry information, to transport information from one area of our body to another in the form of an electrical impulse called an action potential. That's what neurons do. They are the wires to our body. The motors in our body are the muscles, but what makes the muscles or tells them to work is our brain sending signals to the muscles so that they fire in an appropriate fashion. And so in order to do that, they're going to carry an action potential, an electrical voltage down their axon, hit the synaptic knobs, and if you studied the motor end plate videos and how muscle contraction is controlled, the synapse, the neuromuscular junction is what we called it, then you know that there's some calcium ion channels here. When the action potential or voltage hits, the calcium ion channels open, calcium enters the synaptic knob, the little synaptic vesicles in here have proteins that the calcium will cause to bind to proteins in the cell membrane here, and all these little bubbles of membrane would end up binding to and releasing neurotransmitter into the synaptic cleft. At a neuromuscular junction, I would have the motor end plate there with some acetylcholine channels, and the neurotransmitter would be acetylcholine. But in this diagram, we could have a whole other neuron here with all of its dendrites, and along its dendrites, it's going to have all these receptors for neurotransmitters and a soma, and then it will go off and communicate to another cell somewhere. So how neurons communicate is sort of this electrical, chemical, alternating. So the action potential travels down the axon, causes the neuron to release a chemical called the neurotransmitter across a small gap called the synaptic cleft. The neurotransmitter would bind to receptors on the next neuron, and then that neuron would fire an action potential. Now, because of, or it may not fire an action potential, we're going to see how action potentials can be created or stimulated to occur, and how they can be inhibited or prevented from occurring. But nonetheless, once you understand one synapse, like the neuromuscular junction with acetylcholine receptors and acetylcholinesterase, the enzyme that breaks down the neurotransmitter, you understand all synapses. They all work the same way. The name of the neurotransmitter changes, the name of the enzyme that breaks down the neurotransmitter changes, and the name of the receptor changes. But ultimately, they all function very similarly. We're going to look at that in some detail in the future. And then once this neuron fires, if it does, it will communicate to the next neuron and the next neuron. If eventually it might go out to a muscle cell, or it might be coming from the receptors into my CNS through the afferent division and ending up synapsing on my brain. Nonetheless, that's how neurons work. What our next video is going to do is it's going to look at ion channels, and it's going to look at how these ion channels are distributed throughout the dendrites, the axon hillock, and the axon, so that we can carry action potentials or voltages down the neuron. So this is a rather simple and quick video, but I wanted to knock out different neuron structures and talk about how the different neurons are shaped, and then we're going to look at this multipolar neuron again in the very near future. So I hope you learned something. I hope you had as much fun as I did, and I hope you tune into more videos. So thanks for watching. See you on the flip side in the next video.